Okay, so far we talked about the energy mediated internalization of the particles or foreign objects within the cells. As you remember here, we talked about the conversion of ATP into ADP, which provides the energy for the cell to form this vesicle, and then the use of dynamines to close this vesicle here and internalize them within the cell. But apart from this energy mediated internalization, we have the passive mechanism, which there is no activation energy. It is only based on the electrostatic attractive forces between the particles and the cell membrane for the internalization. As you know, the plasma membrane, the phospholipid membrane of the cell, is negatively charged. And if we have our particles with the positive charge, which come into the vicinity in the short distance of this cellular membrane, the electrostatic forces, the attractive forces here will make the particles to stick onto the surface of this membrane. And now the interaction of different forces, as you see here, we have the electrostatic forces, we have the surface tension forces, and the forces of the actin fibers, which are polymerized here. And if the net interactive force here is in a way which can internalize the cell, so I mean if the electrostatic force is the dominant one, we can make sure that the particles can be internalized. For example, in this study, you can appreciate the analysis of the passive internalization of quantum dots. We have the quantum dots here within the red blood cells, and that's it. Well, these two diagrams I'm sorry, want to show whether the particle internalization is passive or energy mediated. Okay, we told you about the conversion of ATP into ADP here, and this conversion can be stopped at lower energy. As you see here, when the temperature is dropped down to 4 degrees, the conversion of the ATP into ADP is stopped, and if under those conditions the cell internalization is stopped as well, we will realize that this has been an energy mediated internalization, because now with inhibiting the conversion, the energy conversion, it has been stopped. Okay, we can also use another inhibitors, as we mentioned in the previous video. Here I mean the dinosaurs and other inhibitors, to see the preferential internalization mechanism used by the cells.